Good afternoon. It gives me a great pleasure to introduce Professor Sir Walter Bodmer, who is one of one well-recognized human geneticist. He wrote a famous book on human genetics back in the 70s, which is to us like the Bible, I would say. He discovered the HLA locus, and then his work in HLA and nickel equilibrium is forming the basis for whole genome-wide association studies. He has been director of the ICRF, president of Harvard College in Oxford. He's currently professor of genetics at Stanford and Oxford, and he has been president of the Human Genome Organization, the European Association for Cancer Research, and many other important institutions. He is fellow of the Royal Society, a foreign member of the National Academy of Sciences. He was knighted by the Queen in 1986, and he has over 20 honorary degrees. In summary, I could spend hours speaking about what Sir Walter has achieved, and we are really honored to have him here in Davis as one of our historic lecturers. So I'll leave it to Sir Walter.
It's a great pleasure to be here and an honor to follow so many distinguished speakers in this series. I was asked when I was last here, and I couldn't quite remember, but I do remember that just over 50 years ago I was once offered a job here. I suppose mistakenly I turned it down. But it is a great pleasure to be here and to meet many of the interesting scientists working here. So what I'm going to talk to you about is, as indicated in the title there, is really eventually with some background to talk about a study we've been doing for many years now on the British population and the way that one can use genetics to really find out a remarkable amount about people's origins and relate it to the history. And I want to start with a little bit of background. I'm sure many of you have given blood samples and you all know about the ABO blood group system. Does that work? Maybe I'm pushing it the wrong way. And actually ABO was one of the first Mendelian polymorphisms, if you will, to be described in humans in 1900, although it wasn't really appreciated at the time by Landsteiner, remarkably, that it was a simply inherited Mendelian dominant. And it was left to the Hirschfelds, a couple who did a lot of the early studies that showed that you could use genetic markers to distinguish different populations, and they first pointed out that ABO was actually a Mendelian inherited trait, and then did a worldwide study. You can see English, rather crude definitions, French, Italian, Germans, all the way to Negroes, and what's it say, Americans and Indians there, Jews were categorized. And what they showed, this was actually published in 1919, just after the First World War, was the first population genetic study in humans, you could say. And they found that the frequency, they just talked about A and B at that time, they were thought to be two loci, and they said the frequency of B varied from relatively low, as we know in the English population correctly, to being relatively high in Asia. And they said, well, it's clear that the population of humans is divided into two groups, the A's and the B's. And they thought that was a fundamental division, which of course we now know is wrong, but it was a very early example of the extent to which one could look for variations in gene frequencies between different populations. Now, many of you may perhaps recognize who that is. That was my great teacher, R.A. Sir Ronald Fisher, one of the three founders of population genetics, together with J.B.S. Haldane and Sewell Wright, and a remarkable scientist in many ways who first showed the compatibility between the ideas of quantitative genetics that came from the biometricians and Mendelian inheritance. He wrote a remarkable paper in 1918 on that. He stimulated my interest in, as you've heard, in the HLA system by his remarkable incisive explanation of the rhesus blood groups as a set of linked loci. He was almost correct to the molecular level. He thought there were three, and we now know there are just two. And in his book, which anyone interested in population genetics and evolution should read, The Genetical Theory of Natural Selection, in many ways a real successor to Darwin's evolution book. He just in a paragraph or so described the field which later I and many others have worked in on the way that selection could influence the interaction between two linked genes. He was a truly remarkable person who actually stimulated a lot of the development of blood group research in Britain in addition to his theoretical work. And amongst his interests was the use of ABO gene frequency data in the UK to try and say something about the origins of populations. And I've illustrated that here. Because during the Second World War, because of the importance of blood transfusions, there was a very large amount of data, even by modern standards, obtained on the ABO gene frequencies by, of course, the classical serological techniques. And he says, interestingly, contact with the neighboring countries was extensive, and he points out that one would have expected similarities as a result of that. And he showed that when you looked at the ratio of, where is it, Scotland up here, of A to O, it was quite a bit different, many more O's than A's in Scotland than elsewhere. And he said he couldn't easily explain that by relationships with the surrounding countries. It was really a forerunner of what one can now do in so much more detail. An example of this quite a lot later, again with ABO blood group frequencies, was a study done by a geneticist, Morgan Watkins, in Wales. And it was again based on very large amounts of data looking at the frequency of blood type A. And this shading illustrates the relative frequencies with a comparatively high frequency indicated by this full shading there, in a part of Wales, which if you know something, and many of you I'm sure won't be that familiar with the history, is actually often called Little England beyond Wales. And it was actually an area that Henry II settled with Flemish farmers who came from over here somewhere and who clearly had different A frequencies. And that really matches, and I'll say a bit more about that in the results I'm going to tell you about, and really showed a relationship between a very specific piece of history and the likely genetic makeup of the people here, where it was known that they kept to themselves and kept out the unruly local Welsh. So it's an example of the sort of detail that even with one genetic marker, sometimes you might be able to say something about. But the development of this sort of work, of course, came with the much more extensive availability of polymorphisms, of genetically determined, well understood genetic differences, notably the blood groups. And my colleague Luca Cavalli-Sforza, you just saw an example of the book that he and I wrote, which was actually published in 1971, 2 years after we finished it, but is still perhaps occasionally worth looking at as a historical object. But what Luca Cavalli-Sforza and Anthony Edwards, another colleague and friend, did was produce the first evolutionary tree, phylogenetic tree, based on the use of genetic data of human populations of different sources, using the then available limited amount of data on blood group gene frequencies. And they devised, that was Anthony Edwards' contribution, approaches to doing phylogeny with frequencies which are now very widely used on the basis of much work in this area that's done using these approaches. And really the tree that they obtained was not bad when you think about the limited availability of information at that time. They did put the Bantu and the Ghanaians together. They put the Australians together with New Guinea. They had a reasonable geographic spread. They did put the Lapps with the Turks and the English as sort of in the middle there. When you consider the limited amount of data, it was quite remarkable. But of course it was looking at population differences at a very crude level. And one of the developments that began to change this a lot was the very thing that's already been mentioned, the development of the HLA system, which is a very complex genetic system which was originally discovered in the search for matching people for transplantation, because of the fact that inherited differences between people lead to rejection, and the major differences that do that are the differences of the HLA system. So I was fortunate enough when I came to Stanford in the early 1960s to collaborate with Rose Payne, who had a lot of material, and I helped with the data analysis, to be involved in the initial description of the HLA system, which was by serology. It's as if instead of just two types A and B, you had a large number. And each of these is really a reflection of a different allele, in this case at the A locus, and then there's a B locus. Each letter and number is a different allele, but described by the serology. And at that time, there seemed to be 6 loci, and it was clear that they were very closely linked. And there was extensive data then that showed that if you match people for these types, especially within families, it really did improve graft survival, and that's still the basis for bone marrow transplantation between HLA identical SIBs. So actually that gave a large amount of new data that could be used to refine population relationships. Just to comment on developments, before I do, very importantly, one of the earliest things that was done with HLA, because through Hugh McDevitt in particular at Stanford, with whom we collaborated, there was the suggestion that the HLA types might have some influence on immune response and autoimmune disease, of looking for associations between the HLA types and disease. And these were quite remarkable. The strongest association, still one of the strongest, with ankylosing spondylitis, a form of rheumatoid arthritis, if you will, of the spine, where in a typical European English type population, the frequency of people who have ankylosing spondylitis and who have this particular marker B27 is close to 100%, where the average frequency in a population would be somewhere between 5 and 10%. And there were several diseases found, of which ankylosing spondylitis, psoriasis, celiac disease, and rheumatoid arthritis remain of great interest and strongly associated with autoimmunity. And hemochromatosis, the one example which actually then led to the discovery of a very nearby link gene, a very interesting story that affects iron uptake. And so that this was an example of the use of a marker for identifying something due to a variant at a linked locus. And it's in fact the interpretation of HLA and disease associations in terms of linkage disequilibrium that has led to all the work that is now going on in looking for disease-associated markers as a basis for studying susceptibility to a variety of chronic diseases. So just to explain this a little, the phenomenon of linkage disequilibrium is quite fundamental to this. And let me try and explain it in simple terms. You've got these two HLA types, A1 and B8, determined by alleles, variants at these two closely linked loci. And you can say, what's the frequency of people who carry A1? What's the frequency of people who carry B8? And what's the expected frequency of people who carry A1 and B8 on the same chromosome? Would be simply the product of those two if there was no association between them. But what you find is a much higher frequency than that which is expected just by multiplying together those two frequencies. And it's that excess which is linkage disequilibrium. The tendency to hold markers together on the same chromosome because they're closely linked, which is a function of time, of the recombination fraction, the distance between the loci, and also possibly selective pressures. So that if, say, B8 was your disease gene and A1 is your marker, you could find an association with A1 that was really due to this linked variant. And that's the fundamental idea between doing association studies for disease. Now, the HLA system turned out to be very complicated. You can't read most of these numbers, but just to indicate that, this is a map of the HLA region at 6p21 up here. This is roughly the position of where the genes are. The whole region is about 4 megabases. And this is the count, the current count, I looked it up just the other day, of the number of alleles, the number of variants at this locus that have been found. It's huge. Similarly for each of these loci, highly polymorphic. For the DR, which these are present on most cells, these are present on a much more limited number of cells. It's a hugely variable system, although the more recently found alleles actually occur with somewhat lower frequency. And that clearly, and there are lots of genes actually in in the middle between these two and between these two. And that shows, because of the linkage disequilibrium between closely linked loci, how if you find an association with one gene here, it may actually, a variant here, it may actually be due to something that's elsewhere in the HLA region held together by the phenomenon of linkage disequilibrium. And that's the whole idea between genome-wide association studies extended to looking at any genetic marker, saying you're looking for those that have a higher frequency in disease than in controls, and if you find that, that's a prima facie evidence that that may indicate where there is variation that influences susceptibility to that disease. And that all came initially from the HLA and disease studies, which still are the most remarkable example of that, of the success of that sort of approach. And just to emphasize that, I mean, the idea is really quite simple. If you think of it here, it's in terms of polymorphism in a gene affecting cancer, that you've got a variation at a locus, and then you get a new mutation that occurs. It's going to occur on one or two of those chromosomes, either the one that carries A or the one that carries A1 or A2. And if it's on the one that carries A1, it'll just stay along with that for a long time and be in linkage disequilibrium, and you'll find an association with the marker here that's probably due to this variation that's happened here. Now, I mention all that in the context of the population studies we've done, because the reason we got the money for doing them is not because we're interested, and a lot of people are, in what we can find out about a population, but because we were producing a set of samples that form an ideal control for disease association studies in the UK. So that's the basis for it, because for those studies to be successful, apart from the careful definition of the disease, you really have to have a good control, especially if your effect sizes, your odds ratios are very low. So what is the study that I'm going to be talking about for the rest of my talk? It's a study that we call People of the British Isles. It was funded by the Wellcome Trust in the UK, a major funder. The aim was to collect 4,000 samples or so, but in a very particular way. Because we wanted to get representative samples from the population, reaching back in time as much as possible, we went only to rural areas, countryside, defined in various ways, but by being not too close to any town that's bigger than about 100,000 people, about maybe the size of Davis or so, and all 4 grandparents coming from the same area, so that they really reflect people who come from the area that you're sampling. We ended up, because these samples have been used in a case control study, large study funded by the Wellcome Trust, we were able to get quite a lot of genotyping done, and for various reasons we ended up by doing an analysis on just over 2,000 patients and just over 500,000 SNPs, 500,000 variants. And actually having a large number of variants is important if you really talk about relationship between population groups that are very close. And so, as I'm going to describe, we did what we consider create a genetic map of the British Isles, and we have produced this resource for future disease association studies, which is being actively used now in these sorts of studies in the UK. Well, just to give some initial data then, each of these spots is a place where the average position of the 4 grandparents of the people that we sampled is. And you'll see that some of them are in the middle of the ocean, but that's because they may have had, some had grandparents here and grandparents here, and they map in the middle. But it gives you some idea of the coverage of the UK. You'll notice that there are obvious holes, like where London is, because we didn't sample in urban areas. We didn't have much sampling in Scotland. We had sampling in Northern Ireland. We were not funded to sample in the rest of Ireland. So we have those data, and we have genetic markers on them, and the question is, what do you do about analyzing them? Now, actually, many years ago, it was my late wife who worked with me and on her own for many years in the HLA system, pointed out that rather than using individual allele frequencies to define populations, if you have data on linked loci, the haplotypes, the combination of 2 variants on the same chromosome, give you much more information than the alleles on their own. And this is extremely important, as I'm going to show you in a minute. So with colleagues, notably connected with Peter Donnelly and Simon Myers, we did an analysis that's based on a published procedure, Lawson et al., and 2012, using a Monte Carlo Markov chain process that essentially was able to match individuals to see which individuals were closest in terms of their genetic makeup, taking into account the position of the genetic variants we were looking at. In other words, using the linkage disequilibrium information. And in this way, you could define clusters of people so that within the cluster, they were more similar to each other, taking into account this information, than they are between clusters. And you can devise statistical procedures that tell you when to stop in terms of when you're not getting any more information that's going to give you any better value of this clustering as a description of the population. And as I'll show you in a minute, I mean, there were a large number of clusters identified, and each cluster is identified by a colored symbol, and the shape and the color of the symbol defines the cluster. So I'm going to show you now the result of this, which actually we found absolutely surprising and striking. So if you look at this map, and I'm going to go into much more detail in a moment on this, you'll see that there are colored symbols, yellow circles here, blue crosses, red squares, different colored symbols. Each of these colored symbols reflects one of these clusters, identified by the fact that within the cluster there's more similarity than between different clusters. And that information was obtained with no reference whatever to knowing where those people came from. We just did the genetic analysis of similarity and then said, where do these people fall? And it's quite remarkable to see that when you do that, you get such a congruence of the genetic information and the geographical information. You'll get some exceptions. There's a yellow circle somewhere up here. But the tightness of these is quite remarkable, even separating Cornwall here and Devon. And I'm going to go through this to give you some idea of the hierarchy of similarities that come from this analysis. Because what you can do is when you've defined the overall clustering, and we stopped, in this case I think we had something like 20 or so, 20, 30 clusters, by which time if you go much further than that, you don't get any further significance to the clustering. And then you can work your way back and say which was the initial cluster that's most different from the others. And then amongst the remainder, what's the most different. And I'm going to take you through some of those to give you some idea of the makeup of this in terms of different groupings. But before doing so, I just want to point out that if you do an analysis of these data using the conventional principal components analysis, taking each allele on its own without taking any note of the relationship between alleles, you get a mess. This is the same data, same individuals, plotted ignoring the linkage disequilibrium. You get a little bit of clustering in Wales, maybe a little bit down here, maybe here, but you get nothing like the patterning we've seen before. That shows how important the approach to the analysis is, namely taking into account this phenomenon of linkage disequilibrium and haplotype frequencies. So let's then go to these different levels. So the first and most striking level of difference was between Orkney. Orkney is a set of islands off the north-east coast of Scotland and the rest of the country. Quite striking, the biggest difference. Now, the history is very well defined that Orkney was a Norse Viking kingdom from about the 800s or 900s until the middle of the 15th century. And so that clearly, as I will show you, reflects the fact that there's more Norse Viking admixture in the Orcadians, which has been pointed out to some extent before, than in any other part of the British Isles. If you then take the next, and let me just, yes, we're doing an association with the genetic information. We collaborate with Paul Longley, a group at University College, who are experts in studying surnames. And surnames, of course, parallel to a fair extent where people come from. And I show you a couple of examples, and this is one of them. So this is a name, Grendel. Grendel is a very well-known name from Orkney. if you look at the distribution of the relative distribution, intensity, relative frequency of the name Grendel in an source of data in 1881, it's all up here in Orkney and in the northeastern tip of Scotland. And if you look in a census and other derived data in 1998, more than 100 years later, it's still all up there. That indicates a remarkable stability in terms of the distribution of surnames. And in fact, the gentleman whom we did here, this is part of a television program, so I'm actually allowed to talk to people. We discovered you can get ethical permission, doesn't matter if you do things for entertainment. But if you do it for scientific reasons, you've got to get ethical permission that doesn't allow you to reveal to an individual actually what their own data are. But this individual, based on various criteria, was, we reckon, more likely, several times more likely, just looking at the relative frequency of his makeup in Orkney compared to parts of the country that we suggested may represent more of the ancient British fringe. But he actually nevertheless had a Norse Y chromosome type, well-known one, M17, which is found mainly in Norway now and in parts of Russia, showing, of course, what you might expect, that there's a mixture of backgrounds there. And as I'll come on to emphasize later, there was no question of the Norse Vikings just wiping out the local population. Well, if you look at the next level of separation, it's quite striking. It's Wales. Comes out as really quite different, remarkably different, as I'll keep emphasizing. And I'm not going to go through all the stages, but just go through a few, and each time look for where the new colors come. No, before you do that, I've got to give you another example of a surname, okay? This is a surname, David Hughes. If any of you know anything about, well, David is a very Welsh name, and Hughes is one of the surnames that were given to people who worked for Baron Hugh at the time when surnames were first introduced, extremely Welsh. So if you look at the distribution of that name in 1881, you can see by far the highest, these are sort of different intensities, relative frequencies, is here in northwest Wales and East Anglia. And you look at it more than 100 years later, of course there's a bit of spread, but still very much focused on that part of Wales, which is an indication of the stability of populations in spite of what people say. And we had data on the Welsh surnames, and we could say how much more likely was it to find this person's genotype in a Welsh population than in the rest of the British population, and it was hugely more likely. And you know, when we went to people and told them that, they were delighted. If we told them something that they didn't want to hear about where they came from, they were very disappointed. People who wanted to be Vikings with horns on their heads, and when we told them, well, it doesn't really work that way, they didn't like that. Well, to continue with the description, if you look at that picture and then look at that picture, you can see where there's a difference come in, which is an important one. First of all, we distinguish North and South Wales. For those of you who know the map of the United Kingdom, it's the United Kingdom of Great Britain and Northern Ireland. We talk about Britain just to describe the main island. This is Northern Ireland, Scotland up here, finishing somewhere around here, I guess. Orkney up there, part of Scotland. Wales here, Devon and Cornwall and Devon forming the western fringe. And you separate now the two parts of Wales, north and south, and you begin to separate this group, so you separate the bulk of England from the combination of north of England, Scotland and Northern Ireland. Now look again, just look carefully what happens next. Do you see what happens next? Several things happen, right? Because I'm not going one at a time. There's a distinction within Orkney. We first noticed this Westray is a northern island in Orkney. And we first saw that there were obvious differences in frequency there. And then you separate out these northern English from the Scottish. What's happened there? Is anybody? Oh, it's there. You separate out the northern English from the Scottish and the Northern Irish. And then if you go a few steps further, you get other changes. If you look at where the changes are coming, see that change does two things at least. It gives you a group here, which are the Welsh borders. And Wales is separated from England by mountains here, and they were very unruly, and they never really were successfully conquered by the Romans or even the Normans, although the Normans built these wonderful castles that you can still see around the coast. So you separate that out, but you also see you separate Devon and Cornwall, which is quite surprising. And that line of separation is remarkable. We have a place we go to on holiday just about here. That's where Plymouth is, and there's the Tamar there. And that line is almost a sharp line of demarcation. It's quite remarkable to get that measure of difference, and remember, entirely based on the genetics. We did not use the geographical information except to place these markers. And if you do another, then you see something else. You see a division here. Now I was just told, I think, Neil, you said that you came from Hull. That's somewhere up here. We do have some differences up there, actually, but they get to be less and less. But what you also see here, which is quite striking, is you see this little group separating out north from South Pembrokeshire. And remember what I told you near the beginning about the Little England beyond Wales, and I'll come back to that in a moment. And I think that's the final one, yes. That shows here, you can see, maybe you're part of that group there. You can begin to see a difference between east and west. But these groups overlap quite a lot. This is a group that's sort of North Lancashire breaking into Yorkshire and separates out also from the two groups in the Welsh borders. And that's actually, and then another separation that is interesting, there are actually two groups that relate Northern Ireland with Scotland. One of these are these orange symbols. I don't know whether you can see them. I can just about see them up here, and the other down there. So there's one group that lumps Northern Ireland with the lowlands of Scotland, another group that lumps them with the Highlands of Scotland, which actually seems to correspond to a well-described tribe that existed at one time. And it's not surprising to find quite a lot of relationship across that, which is almost the narrowest point of contact, of course, between Britain and Ireland. Now, obviously, there's an interest to relate that in some way to what is known about the history and the archaeology of the British Isles. And the history of the British Isles is relatively simple. It's not been fought over for years and years the way that the continent of Europe has or other parts of the world. And I just want to give you a brief overview of that. So actually, although there's evidence that modern humans may have come to the British Isles as much as 30 years ago, the evidence is that there was no permanent settlement until after the end of the last ice age, which is about of the order of 12,000 years ago. And at that time, there was actually a land bridge. My own view is that this should extend a bit further into the Channel. But there was a land bridge between Great Britain and the continent of Europe. There was not a land bridge with Ireland, nor with Orkney. But it's quite clear that after the Ice Age, people will have come up from various parts of Europe here as the weather improved, and were able to get to the British Isles either this way over land, or possibly already at that time, to some extent, by sea, since the distance wasn't so large. But then, for many years, there was not any other major influx into Britain. Agriculture, which obviously had a huge impact on population size growth, and on the whole culture of countries, came to the British Isles about 6,000 years ago. And if you follow Luca Cavalli-Sforza's ideas, which I think are fairly well established, that actually agriculture came with movement of people from the Fertile Crescent in the Middle East, basically, who gradually moved over, but it wasn't the people that moved. They moved a little bit, they intermarried with the locals, then they moved further, and that led to a gradient of gene frequencies, which Cavalli-Sforza and Ammerman have described. But by the time they got this far north, the differences from the Middle East would have been diluted out, and there's already evidence that perhaps a lot of this movement was cultural rather than movement of people. Remarkably, agriculture seems to have spread throughout the British Isles relatively quickly in a matter of a couple of hundred years, which I find quite surprising, but that's what the latest data show. But after that, really the next most important incursion into Britain was when the Romans conquered Britain around about 40 AD, or 40 common era, as we're supposed to say nowadays. And they came and they settled in Britain. There was a large area here which remained by far the most Romanized area of Britain. They never really managed to conquer the Welsh. They didn't get that far into Cornwall, and if you ever go to England, it's quite a lot of fun. I think it's up here. Where is the Hadrian's Wall? It's somewhere up here. There was the furthest north you could go, and you can still see Hadrian's Wall to this day as the major barrier that separated England from the unruly Picts up there. The next major change, which really did bring people in a major way, was, of course, the invasion of Britain by the Anglo-Saxons. But that didn't come again until after the decline of the Roman Empire in about the 400 AD, and the Anglo-Saxons and the Jutes and some of the Frisians, they came from this area, the Danish peninsula, going along the north coast here through Holland and Belgium and parts of Germany. And they came and they largely settled in these areas around the east, and that was in about 500 AD. And in the meantime, there was probably some exchange between Cornwall and the Bretons and some exchange between Ireland and Britain. But it's always difficult to know how many people were involved in these migrations. And then the last major change that influenced the population was when the Vikings came. And there were two colors of Vikings. There were the Vikings from Norway and the Vikings from Denmark, who had a similar culture but a different genetic makeup. The Vikings from Norway, they colonized Shetland and Orkney and up here, and to some extent around the west coast of Scotland. They did come further down, but didn't settle as much. And the Danish Vikings, you got the Danelaw, which settled around here, but it's not clear how many were involved. And then later, but with very little influence on the population, of course, you got 1066 and William the Conqueror and all that, and that's the last time anyone really managed to invade the British. So it's a relatively simple history, but the Normans, who came mainly as rulers and didn't leave many people, didn't leave many of their genes behind, they actually came from this area, which was largely populated by that time from some of these Vikings. So now look at how this can be related to what we can find out about the British population by taking into account information on the surrounding European populations. So with colleagues, there's an analysis being done using actually multiple sclerosis, genome-wide association data of over 6,000 Europeans from different parts of Europe, indicated here, much less finely defined in terms of their origins, often only by the country or by a hospital or by a large city. But we can use the set of markers, exactly the same 550,000 markers, to do a cluster analysis of these data, just as was done for the UK. So each of these circles represents a location where people were sampled, and the different sectors and their colors represent the different clusters, which already shows you that this is a coarser definition, because if you go to here, which is Stockholm, you get a huge mixture of people, although a lot of them reflect one particular cluster. There's a lot of other clusters involved. And very few of these circles represent only one of these genetic clusters in the way that is the case in the UK. But it does give quite a lot of differentiation. You can see that Italy is quite separate from the others. You get none of these greens anywhere else. North and South Italy is different. Quite surprisingly, because each color again is a different group, Denmark, Norway, Sweden and Finland are quite different, much more different than I must say I would have expected. And then you get 2 or 3 groups in France, and one group which is interesting, which you find probably more in the south of France, that also fits with some of the Spanish samples that were taken, although mainly from Barcelona. Now, if you look carefully here, I want you to notice some of these numbers. 31, I can't read them all from here, 17, 12, 14, 18 is Denmark. There's a number 6 and a number 3 somewhere here. I mention those because then what we did next is we did an analysis that says to what extent can one explain the makeup of the UK clusters by mixtures of the European clusters. And again, it involves a fairly complicated statistical analysis similar to the clustering in a way, clustering between rather than within, so that you can get an estimate for each of the British clusters. If they were made up of the European clusters, what would be the relative contribution be of those different clusters? And the reason I mention those particular sets of clusters or groups in Europe is because we find that only a limited set of those contributed, the European data contributed to the British clusters. Nothing from Italy, nothing from Finland, nothing from Poland, nothing from South Germany, indicating really that in a way is reassuring that the constitution that you get from looking at that contribution from Europe comes from the places where you would expect it to come from. Well, you can then represent the picture in this way, which in a way is almost, after the initial clusters that I showed you, the most important summary of what we've been talking about. So at the level of 17 clusters, each of these is a circle at the place. So it's Devon, Cornwall, Devon, South Pembroke, North Wales, the Welsh borders, London, different parts of the north, the two groups that correlate with Northern Ireland with Scotland, two groups in this part of Scotland, and then three different Orcadian groups. And the sectors represent the proportionate contribution from these different European groups, and each of those are defined here. So those are the Norwegians. If I look at my own picture, I can probably get it better. You've got the Norwegians. You've got a group. Let me look at it here, because I can't read those numbers well, but it's quite important to think about those. I don't know, this thing keeps going into sleep. No, it shouldn't do that. I can hardly read what I've got here, never mind. Anyway, you can see, if I move over here, so that's Norway group. That's the group that overlapped France and Spain. That's one of the groups from France, another group from northern France, a group from West Germany, Belgium, a group from North and Northwest Germany, the pink ones, the red ones from northern France, and the 18 from Denmark. And you can see some remarkable differences. It's very obvious that the Welsh populations are quite different from the others. They have none of the red, none of the pink, and mainly these two groups, which are these groups of 14, the Northwest France and West Germany. If you look at the blue, remember that's Norway, a large contribution from each of these Orcadian populations, as one might expect. A reasonably sized contribution, as you might expect, as you move down Western Scotland, also in North Scotland, and as you move down to the rest of England, less and less, even in Wales, where it's thought they would have settled, until when you get down to this area. And remember, this southeastern group is by far the largest, remarkably homogeneous, and has virtually no contribution from Norway. And then there's some other interesting features. Some of these, these three, this dark green, the lighter green, not very light green, and the yellow, you find these in all the populations. Everywhere you look, you find those, which suggests that they are the oldest contribution. And that fits with the fact that you find them overwhelmingly the highest frequency, both of these in Wales, suggesting that if you want to look for a population that may be the closest to the original inhabitants of the British Isles, it would be these, and it would reflect these contributions. Then if you look at the other side of things, if you look at the pink contribution, which comes very much from the area you might think the Anglo-Saxons came from, you see that's present here, present there, present there, nothing at all in Wales, lower but still significant frequencies here, gradually decreasing until you find practically nothing up here. But interestingly, these two populations, which are very similar, Banff 1 and Banff 2, differ with respect to that Saxon contribution. And then if you look at the red contribution, that's also interesting, because that's from part of northwest France, from where you might have thought there would be a lot of communication with Britain, and that you find not at all in Wales, absolutely not found there, whereas you find it in all the other populations. And that actually suggests to us that there was a group of later migrants than the earliest that we talked about here, but that predate the Saxons and probably predate the Normans, the Romans, rather, predate the Romans, something which had not really been supposed before, but which makes a lot of sense that there would have been continuous movement over from northern France into this country over quite long periods of time, and that simply didn't get across to Wales because of the mountains and the separation of the Welsh groups, and was more highly represented in the south and somewhat diluted as you go north. So you get a remarkable patterning. Now I want to just point out, you remember we talked about Little Ireland beyond Scotland and the difference between South Pembrokeshire, Little England beyond Wales, the difference between South Pembrokeshire and North. And you can see that reflected in the higher proportion of the yellow here from Belgium. And also, although it's hard to see here, a relatively higher proportion from Denmark, which is intriguing when we notice this, because then our archaeological friends said, well, it's well known that there's a tendency for a higher frequency of Danish place names in that part of Wales. So I just want to suggest to you that with this sort of analysis, using a carefully selected population, using a large number of genetic markers, and using an approach to analysis that really takes into account the linkage disequilibrium, the patterns of how variants come together on the same chromosome, you can get a remarkable patterning of what is in fact a set of extremely closely related populations, but one that you can have some confidence in because of the extent to which, where it's known, it matches the historical and the archaeological information. And it says a few things. One, which I think is really quite striking, is it indicates a remarkable stability of populations, much more than is usually assumed. People say, oh, people migrate all over the place. But it's not true. They tend to stay where they are and marry their local folk. Now, we have focused this by going to rural areas, and people, all four of whose grandparents come from those areas. But nevertheless, that stability, and I think of my mother-in-law who came from Manchester, and she went to London once in her life to visit us when we lived there. There is not that much movement around, and you could not see this sort of patterning if there was much more extensive movement outside, of course, the major urban areas. The other thing that's quite remarkable is the relative uniformity. If I really take you back to this picture, this large group here, the relative uniformity of that is quite striking. It corresponds, it means that really with the resolution we've got, which can distinguish even these two populations, we see no differences within that area, which is quite striking. It does correspond to the Romanization of Britain, which was largely that, and suggests that really during long periods of time, and assuming that people do tend to marry others that are close to them, nevertheless over a period of time you get a spread and a uniformity included if there are no real political or geographical barriers. And I think that's quite a striking observation there. So I just want to suggest in finishing that we believe that this sort of analysis, which I think is the first time that this sort of detailed information has been obtained on one population, looking very carefully, could be done in many other parts of the world, and I think would be of great interest, could be done obviously in Europe. We've tried to get money for that so far unsuccessfully. Most interestingly, could be done in Africa, could be done in parts of Asia. So I leave you with that thought. Thank you. There must be some questions. Yes, there's one. Okay. There's one there.
Are there historical events based on historical background, you might have expected to sort of leave a footprint genetically that you found
Well, I've described one.
underrepresented?
Remember, there's very little history that goes back before something like, you get some history from the Romans, you get some history from the Venerable Bede about 500 or 600 or a bit later AD, but very little written history, and what there is not very reliable. But of course there's documentation of the Saxon invasions, we know about that, but we don't know just how many people came. There's clear documentation of the Norse Viking invasions of Orkney. I've shown you the example of the Little England beyond Wales. So these are very specific examples of the correspondence between historical events and what we can show genetically. Yeah.
You said that there are little Italian in this population despite the Roman colonization. Does that mean the Roman colonization was mostly a cultural event?
The question is there any evidence for Italians because of the Roman occupation? The answer, first of all, is simply there's no evidence for any Italian admixture. But the second answer should be that the Romans were not all from Italy. In fact, most of them were not from Italy. They were from all over the place, a lot of them from Middle Europe. A lot of them, most of them left, some settled. By the time the Roman occupation finished, there were already some Romanized Britons. So, you know, at that time it was thought the population of Britain was perhaps of the order of 2 million. Even if 100,000 people stayed there, you wouldn't see that, because it would be spread all over. So there's really no evidence for that. Normans are very hard to distinguish, because they're so similar to the other groups that came from the same area. And until we have more detailed information on those parts of Europe, it's really where you need it from is Denmark, Holland, where we didn't have any data, Belgium, and the north of France, collected in the same way as we did in the UK. Yes?
When you select a population, don't you find a difference between the different groups that you selected? These are not random. These don't represent the population of the area that you presented. You presented a subgroup of that area. So you did a good selection.
No, let me explain. We selected those people on purpose. We purposely selected people that represent what would have been there maybe 100 or more years ago, because that reflects what really tells you about the origins of the British people. Right? And everybody else who's British is made up of some mixture of those. So the way you can deal with the problem that you want, if you really want to be fussy about controls, is you could match your controls from the populations we have to match the samples you've got for a disease. And maybe that would be necessary, and if you're really interested in odds ratios of 1.1, which everybody's finding now, you might have to do that. But the whole aim of this was not to do that. If you just go into the middle of London or Manchester and even ask everyone who's British, you won't find anything out about their origins. You won't be able to describe their origins without having the data that tells you what their origins really were by sampling the population more carefully.
But the rural people are the ones that are more stable, that are attached to the land, are the ones that move the less.
Well, of course they're the ones that haven't moved. They therefore represent the place from where they come. If you're trying to say that only more intelligent people move to the cities and that's connected with our genetics, I think that's probably unlikely. It's quite clear that the major movement has always been the urbanization happened during the 20th century, basically, when you went from 75% or more rural to 25% or less rural, and even now in the rural areas you get people like me living there, and I have practically no British ancestry at all anyway. So I think the only way you can really describe a population is by finding out where it comes from, and then you can reconstruct mixtures. And I think the most difficult population in the world to deal with is the United States of America, because to study that, you've got to study every different population in the world. Yes?
Could you potentially use mitochondrial or Y chromosomes to see about different movement patterns of
Yes.
men and women?
Yeah, we have data On the mitochondrial DNA and on the Y chromosome, we've not yet done a detailed analysis. You could find out some things. We have some evidence about that, about differential movement of males and females, and that is obviously of interest and something we still plan to do. We've not had time to do a lot of the more detailed analysis of specific markers that may, for example, be subject to selection. You can look at all the skin color markers, for example, a variety of other things. We're also doing now a study of the genetics of the face. When we find the genes variants that affect that, we'll look at their distribution as well. But the notion that you can easily tell people's origins from Y and mitochondrial chromosome DNA, which is widely touted by some of the companies that do that, has to be fundamentally wrong, because there's too little information, especially the mitochondrial data is very limited in its variability within Europe, let alone within the UK, and is not a very good marker of origins in this way. You can only get this sort of detailed information if you look at a much wider range of markers, and you're then looking at the average over a set of markers spread throughout the genome, and if you find particular differences for a given marker from what that average shows, then you can begin to say something about that, whether it's due to selection, differential migration, and so on. Yes?
There's also considerable linguistic variation in the different groups. And if you superimpose a linguistic analysis of word usage and where that traces back over the genetic profile, does that match? I mean, is there sort of the cultural history reflected in Welsh, in Scottish?
Well, yes and no. I did actually show, but I didn't mention it, that one of these pictures actually mentioned, I think it's this one, the different languages, the different Celtic languages in different areas. I think it's a very interesting question, and various people, including Cavalli Sforza and Mark Feldman, have talked a lot about the relationship between genetics and language. As far as I can see, if you look at a broad context, you'll see something. As soon as you get into more detail, you lose it. And I think that's particularly striking when you think about this uniform group here, and I've recently been talking to some colleagues who are interested in British dialects. And it's quite clear, and I know it just from local, anecdotal knowledge, that within this huge area, which is relatively uniform genetically, you've got a variety of different dialects. You get a West Country dialect that extends here, you get Cockney, you get dialects that correlate with East Anglia, you begin to get up here some of the northern dialects. So what that shows is that when you get down to that detailed level of language, you seem to lose the correlation between language and the genetics. Now, whether if we had additional data and could say for each of these groups what was their predominant dialect, would that begin to give us some breakdown within here? That's possible. But I think that shows that you can get differentiation within groups, and you get that to some extent within these groups here, that are extremely similar, but they're still marrying locally. And even if you get a few examples, like we obviously got some examples of people here that went there. Well, there were Cornish miners that went to mine up there in the northeast. And you might get a situation where a whole family moves there and they tend to marry within each other and not to marry with the local different people. So you can get these phenomena that actually begin to distinguish quite starkly a cultural definition and a linguistic definition of a group from what you get from the genetics. And I should have emphasized that the genetics tells you about the whole population. The history and the archaeology usually tell you about the leaders. The conquerors tell you about people who are historically significant and are not necessarily representative in any way of the population. That would be true if you took the Normans, for example. So you can get a huge cultural impact called elite dominance from a relatively small group of people that actually then doesn't match what subsequently happens genetically. Yes?
You just alluded to facial features.
Yeah.
I wondered, can you allow any other morphological characteristics of diseases that tend to associate with these?
Well, I specifically left out a slide of what we're doing. In fact, for the last few years, our major interest has been in studying the genetics of the face. But we do it in a way that I think is totally antagonistic to what you're suggesting. I think it's the wrong way to think of the average face of a given group of people from somewhere. Faces are enormously variable, almost totally genetic, as you know from the similarity of identical twins. Probably, I think, influenced, and we're beginning to find evidence for that, quite strongly for particular genetic markers. So I think when you talk about a face from a certain place, you're talking about the relative frequency of that type of face there. And that is something that I hope we will be able to look at. And it may be that as we describe the genetics of facial features, we'll get very good markers that begin to distinguish populations from each other. Because, you know, you even think you can distinguish a Frenchman from an Englishman. They're separated by 20 miles of the English Channel. Because almost certainly facial features have been very important evolutionary, special regions of the brain for recognizing faces, membership of the group, almost certainly some sort of associative mating connected with facial features. So those are very interesting questions, but I think until we really understand the underlying genetics of facial features, we won't be able to answer them properly. Yes?
I have a question. This is a very powerful analysis, and I'm wondering if you used a very careful, you're using a very general population, but if you used a very specific population, like a selected population, like Australia, where their population was carefully selected by judges, about 1700, I guess 1720 or something like that, and you used the same analysis, what do you think that would look like?
I'm not sure I know how to answer that question. By some criteria, ours is quite a selected population. I think you're beginning to relate to the possibility that, you know, the people that happen to be committed to going to Australia were criminals, and maybe they're genetically different from the people that didn't go there. I think that's a little unlikely, but otherwise it's not clear. I mean, I don't think the judges obviously selected on the basis of knowing any genetics. There is, of course, a long-standing notion of what's called physiognomy. I have a lovely book that was given to me by an aunt of mine written by Gargold Labatier, with the most beautiful pen and ink descriptions of faces. And he made his money by telling the leaders of his country whom they should employ for what purpose based on facial features. And there's a lot of thought behind that. And there may be some evolutionary arguments for being able to recognize something in a face that you choose as your mate. I think that's still hugely speculative. But if there were anything like that, the only way we'd find out is by determining the genetics of the face.